but unfortunately, this is not in my hands. It's good to know, but based on your previous answers, I really advise you just have a look at YouTube videos. Okay, so I think that's it for today. I have a good, thank you very much for your feedback. I have some information to work on, and it will be good to move now to the lectures. I'll stop sharing this one and I'll go to the PowerPoint. Okay. So today's lecture is gonna focus on how do we isolate DNA. But since the process of isolation of a DNA is quite similar at certain aspects with the RNA, I kind of blended together this lecture so that I hope by the end of this lecture, you will be able to understand anything related to the protocols that are used for DNA isolation or RNA isolation. So when you get to a lab and you want to perform an experiment on proteins, so you have to isolate the proteins or the DNA or the RNA, you have to have the basic knowledge of how do you handle the DNA, how do you handle the RNA, do these samples have to be all the time on ice? When do I put them in -20? When I put them in -80? What are the material that I'm using? Is there any material that I'm using during the extraction process that is risky to use? Safety measures to be taken into account. So I will mostly focus in this lecture on the nucleic acid isolation. In the next couple of lectures, I will cover the restriction enzymes and the ways, the techniques that we use to separate the DNA on a gel or for sequencing and for some downstream applications. Okay, so if you come to a lab and you are performing an experiment, you have to first look for the strategy that you need to follow. So imagine you have some cells, and the aim of the experiment you want to do is to extract the nucleic acid from those cells. So you have a plate that is cultured cells. And to extract this nucleic acid, you have to have in mind what will happen in every step so that something goes wrong, you can correct it, and say that you are performing the protocol, you would know what exactly is needed to succeed in isolating a good quality DNA that you can use in downstream experiments. So if we lyse the cells, and as for now, I think we're able to lyse the cells because that needs a lysis buffer and some sort of disruption that we discussed in the previous lecture in the fractionation process. But if you look at this cell here, which is presented in a schema, it shows you that if we lyse this cell, the nucleic acid in the cell, they are in compartments where RNA and DNA are together. So in other words, if I lyse the cells with a detergent and mechanical breaking, the RNA, the DNA, the proteins, they will all form one mixture. And that single mixture is going to be difficult to dissociate. And therefore, you have to put the strategy, what exactly you need to isolate from this experiment so that you know what you have to eliminate. So the isolation strategy must take into account first the buffer selection, and then the biochemical property of the molecule that we want to separate. In this condition, it's going to be the DNA. So if I'm just like today, I come to a lab and I'm putting this strategy, I would say I'm isolating the DNA. So therefore, I will need first to solubilize the cell and remove the membranes. And this is the cell lysis step. This should be followed by protecting the DNA from degradation, because, for example, in our cells, there are DNases that can degrade the DNA. So when you make this mix here after lysis, probably those DNases can destroy your DNA. So you want to protect your DNA. Then you want to get rid of other material that is like making contamination for your DNA, which is mainly RNA and proteins. And then eventually you want your DNA to go to a fraction where the majority of that fraction is in fact DNA. Then you would like to recover that DNA from the solution, because if it's soluble, you want to precipitate it. And when you precipitate the DNA, most probably you will get some impurities. So you want to wash the DNA, so you remove most of the impurities away. And finally, you will get a pellet of DNA that you can resuspend. And you can resuspend in a buffer that you can either store or you can use in the downstream applications. If it's a cloning or it's a transfection, we will come to these applications down the road. And obviously, you have to take a good caution about the toxicity and the volatility of the compounds you are being used for your health and safety. Okay, so that's how I figure out a very simple strategy there, how I will isolate the DNA. Now, obviously, this is a very, very good studied domain, and there's a lot of protocols that has been published over many years to isolate the DNA. So we are not inventing these steps, but I'm walking you through these steps because it's important to understand what each step of this mean and how we do it. So first, the biggest problem we have is that both DNA and RNA have similar structures. So the RNA, they're mainly based on the ribose as a sugar moiety, while the DNA is using the deoxyribose as the moiety of sugar. Now, if we look at the property of the carbons on the ribose and the deoxyribose, you can see that the carbons 5, 4, and 3, and 1, they have more or less the same properties. The only difference between the ribose and the deoxyribose is the OH group at the 2', the carbon 2'. Now, the directionality of the RNA and the DNA, the one that we talk about in all the textbooks in genetics, the 5' to 3' end, is maintained. And once new ribose or deoxyribose is added to the nitrogenous bases of the DNA or RNA, it will generate what we call the phosphodiester linkage. And therefore, the phosphodiester linkage is negatively charged. So at the level of a neutral pH, both RNA and DNA are negatively charged. And therefore, at this pH, both RNA and DNA are soluble in aqueous solution or in aqueous phases. So this is important to know, because this OH group renders the RNA more polar than the DNA. And in other words, it makes the RNA more stable in acidic conditions, and it renders it more soluble in acidic conditions compared to the DNA. And this is the property that we will use to dissociate RNA from the DNA. So I wrote it down here for you. So since RNA is more soluble in acidic pH, lowering the pH of the extraction buffer can lead DNA to go into the organic phase, or in other words, it precipitates out from the aqueous phase, while RNA will be retained in the aqueous phase. So this is the basic concept how we separate RNA from DNA. Of course, this is more complex. I didn't add all the details for you here. There is tons of research papers about the properties of RNA in acidic solution compared to DNA and how that precipitate and RNA becomes soluble while DNA precipitate. So if I take this into consideration, then I try to find what is the most suitable buffer or solution in order to isolate the DNA or the nucleic acid. And the one that people are using since long time is the phenol chloroform mixture, where phenol is known to be an aromatic organic compound. When mixed with chloroform, it will make this solution, which specifically denatures proteins. However, the nucleic acids are still intact. So this is already very good, because if I go back to my slide, you lysed your cell, and then you want to protect your DNA, and then you want to degrade RNA and proteins. And by using phenol chloroform, you already got rid of the proteins, because most of the proteins are denatured. You end up with a situation where you have DNA and RNA together, and we can deal with this down the road. So that's a very good solution to use. Now, depending on the phenol we use, we can decide if we extract all DNA and RNA together or only RNA. And as I said here, it's the pH that dictates whether DNA and RNA are isolated together or not. So in the conditions where I want to isolate all the nucleic acid, it's important that I maintain the phenol at a pH of 7 to 8, a neutral pH. Now I will have RNA and DNA together. Okay? And both RNA and DNA will be separated to the aqueous phase, where the organic phase contains the proteins and other components of the cell. So specifically speaking, if I would like to isolate DNA, the pH has to be maintained at 7. And this is mostly by using a tris or tris EDTA component of the phenol. And in this situation, the DNA is not going to be precipitated into the organic phase. It will stay both RNA and DNA in the aqueous phase. Now, in conditions where I want to isolate only RNA, I can simply shift the pH from 7.8 to 4.8. And in this situation, the DNA will become more enriched in the organic phase, and in the aqueous phase, I will have mostly RNA. And that's a way to separate RNA from DNA. Now, let's forget about RNA isolation itself. Let's consider we are isolating DNA today. So we have the phenol, which is 7.8. Therefore, I have RNA and DNA both in the aqueous phase. Protein and rest of the component went into the organic phase of the phenol chloroform. So what would I do next? I would get rid of the denatured proteins, and this would be a process of centrifugation in order to separate the aqueous phase from the organic phase. And in order to get a very good separation, we have to use chloroform. So whenever you use phenol and you see that there is a usage of chloroform, the reason why we use chloroform is that it has a higher density than phenol. Therefore, imagine you have an aqueous phase and an organic phase, like water and oil. To have a very good separation between the two is very difficult. If you add a chloroform on top, the chloroform will make the separation much, much sharper. And therefore, the aqueous phase will be very nicely separated from the organic phase if you perform centrifugation. So that's the main reason why we use chloroform in mixture with the phenols. The other thing is that the organic phase sometimes makes foam because there's a lot of lipids that's going to go into that phase. And in order to avoid foam formation, we can use the alcohol. So in this condition, the isoamyl alcohol. So whenever you see this component, phenol, chloroform, isoamyl alcohol, it would be for DNA isolation, having a better separation of the phases and avoiding having any foam formation in the organic phase. In addition, if we are using this compound, this mixture, sorry, on the 7 to 8 pH, then most of the RNA will, the RNases will be inhibited, so that the RNases are in fact enzymes, right? So the proteins will be denatured, and therefore most of the RNases are inhibited. And smaller size RNA will be also kept into the organic phase. So at least by these couple of slides, you already know if you have two types of phenolic solution, one of pH 7.8 and the other 4.8, you directly know which one you use for DNA extraction and which one you use for RNA extraction. And if those compounds contain chloroform or isoamyl alcohol, you would know why those compounds are added. And in case you are not getting a very good separation of the two phases, you will directly check if you have the chloroform and isoamyl alcohol in your solution or not. Okay, now imagine we do the disruption. And as I explained in the previous lecture, that the disruption itself by mechanical ways or non-mechanical ways is not only enough for the isolation of macromolecules. In this particular lecture, we are doing a combination of mechanical, which is homogenization, and the chemical lysis, which is a lysis buffer followed by phenol chloroform, in order to isolate a nucleic acid from cells or tissues. So this is a practical example for you to show you that we have to combine these disruption methods in order to get macromolecules like DNA or RNA for downstream experiments. So these are the phases that we were talking about. If you perform the steps as I explained, then you centrifuge, then you will have this upper phase, which is the aqueous phase. Most of the DNA is there. DNA and RNA are there in case of 7.8 pH. You have a separation of interface, which denatured proteins mostly stay there. And the rest of the proteins and other components would be in the organic phase down there. Now, sometimes you have this in like kind of pinkish color. So the separation will be, this is a white and this is a pinkish color after separation. Now, after lysis, like let's imagine here we have chosen the lysis buffer. We disrupted the cells with whatever mechanical homogenization. Then right during the lysis, we have to have detergent in order to get rid of the membranes. And this could contain any of the detergents we just spoke about before, like SDS or MP40 or Tween 20 or whatever. And or lysozyme to destroy the cell walls. Now, within those two, the lysis step, we have to take into consideration the treatment with RNases, because we have to treat, if we are isolating the DNA, we want to get rid of the RNA. So in order to get rid of the RNA, after we lyse the cells and all the RNAs are within the mixture, we simply add RNases. And by digestion, over an hour or so, you will get rid of all the RNA. Now, we also do assuring steps to remove any remnants of the proteins and most of the protocols, if you see proteinase K, the proteinase K is used for the digestion and denaturation of the proteins. So these two steps has to take place before we separate the mixture into aqueous and organic phase. Then we add the phenol chloroform and we perform the centrifugation, and therefore you get this separation very nicely as I explained here. Okay, so now I would go to this video. It's around few minutes. I'm not sure if I'll stop sharing that one and I'll go to that one, share. And now it will make a lot of sense for you what I was explaining.
Pepperoni pizza for only $7.99 every day. Hot and ready.
Okay. So at this step, they are lysing the cells. You have the cells, you collected them, and you added the lysis buffer. And you resuspend, so you mix the buffer around the cells very well. Then you add the RNase A, which will degrade all the RNAs. Obviously, you have to incubate for the right time and temperature, so the RNases degrade all the RNA. Then you are adding proteinase K, so you are destroying any proteins. And this works at higher temperatures, like 50 degrees. And now you're ready to do the phenol chloroform isomile. You added the chloroform, phenol chloroform, and you do the separation by centrifugation. After separation, you got the aqueous phase, which is on top. And that's recuperated. And you can see the white interface. You want to avoid this. So the nucleic acid are already on the top, or DNA, in fact, because the RNA is degraded. You can repeat this many times to get a better quality. So they are repeating this step one more time. Obviously, you need to go again to centrifuge to get the aqueous phase and the organic phase separated. And the aqueous phase now contains pure DNA. The next step, so they are doing this 2 times, and now they are adding sodium acetate, which is high molarity, 3 molars, and this will cause precipitation of the DNA. If you mix and then centrifuge, you will get a pellet which contains the DNA. And at this step, you are washing to get any impurities in the DNA. So you simply add ethanol, 70%, and you repeat the centrifugation step. And after centrifugation, you will get the pellets. Most probably you cannot see them by eye unless it's big amount. You keep the DNA to dry, so you don't want to have any remnant alcohol there, ethanol, and you simply dissolve your DNA. Okay, so let me answer the questions you have. A buffer and detergent can be interchangeably, right? The buffer contains detergents. A buffer must have some sort of detergent like SDS or 20 or stuff like that. Would non-ionic detergent be preferred source here to prevent denaturing of nucleic acid? So the non-ionic, I think in this condition, most of the time we either use the MP40 with a very little amount of SDS. I agree with you. Most of the time we use those. In the next couple of slides, I have some more examples, which will show you some protocols where we can separate all proteins, RNA, and DNA. Okay, so let me stop sharing this and go back to that. Okay, so in case you run into a lab where you see, most of the time you will see protocols that you get from companies which would sell you the phenol chloroform, they will list those steps for you in a way that it tells that first you have to homogenize the tissues and you have to make sure that you are at 4 degrees in a sterile environment, depending on what is the downstream experiment. So for the homogenization process, you can check what we did in module 3. So then the lysis process, where you use the detergents. I don't know what happened. Okay. Can you see my screen? For some reason, I can't. It says the sharing stopped.
We can see it now.
Okay. Yeah. So then the next, okay, come on. So after the cellular lysis, you have to make sure that you are doing an RNA degradation step. And that's what you saw on the videos. They first lysed, then they added the RNA degradation by RNases. Then you want to preserve, of course, your DNA. So you inhibit DNases and mostly by EDTA, and that's the factor they use. Then you break down any proteins with the proteinase K. Then you added the phenol chloroform. You got the separation into 2 phases after centrifugation. You removed the salts by washing, and then you precipitated the DNA, and then you resuspended the DNA. So these are basically the major steps that you perform in order to extract DNA or RNA. I would stop here and I would take your questions before moving for the next couple of slides. Any questions? Okay, sounds like it was kind of clear. Okay, so in this slide, I summarized everything I talked. before. So first cell lysis, then RNA degradation, then protein degradation, then denaturation of the proteins by adding the phenol chloroform mix, which is, that's the mix you have to be careful. It's phenol chloroform isoamyl alcohol. Then you would do the centrifugation, you get the separation into two phases and an interphase. After that, you will take the aqueous phase where the DNA is, and you will recuperate it. You will wash it. First, precipitate it by adding sodium acetate, and then wash it and resuspend it. And you will finally get to the final DNA isolation. Okay, so in this setup, it was meant that I want from my cells only DNA. I don't care about the proteins and about the RNA. And this is an old protocol. And the reason why I went through this protocol, I really want you to understand things, not memorize it, or not see it without knowing what it is. Okay, I have somebody who's asking a question.
I just
Yes?
wanted to ask how many times you would sort of repeat these steps in order to get the best results. Like in the
Yes.
video, they only did it twice, but how many times can you do it before it might, I don't know,
In
damage?
general, one is enough if you do it perfect. 2 or 3. Like, I will show you examples that the more you do it, the better.
Okay,
Like,
thank you.
some protocols go 2 or 3. Yes. Okay. So now, instead of doing this laborious protocol where I have to do all these steps, companies came out with novel protocols, and I'm 100% sure if you go to any lab, they will have these spin columns. These spin columns, they have a silica, which is a matrix that has some co-tropic agents that been added. And these agents will specifically bind into one type of macromolecules, either RNA or proteins. So this is a type of separation of DNA or RNA by using a solid phase extraction method. It will save you a lot of time, but still the basics of the technique is still the same. So what you need to do is first to lyse the cells or the tissues that you are using with the lysis buffer that they provide you that is suitable for those cells or this type of cells or tissues. And this lysis buffer most of the time would need to be some sort of mechanical disruption. So you would use a homogenizer with the buffer that comes with the kit that contains detergents and enzymes. Most of the times they wouldn't tell you what are the enzymes, but you would know right now that it's proteinase K or cellulase. After the lysis step, instead of doing the phenol chloroform extraction, what you have to do is simply pass it through over that column. So you fill it here and you centrifuge and everything passes through. And these tubes, they have these collection tubes. So you put it inside during the centrifugation. So everything passes through except the molecule in question. So in this situation, for example, if we have the guanidinium hydrochloride added to the silica, DNA and RNA will bind there and everything will pass. So the next step would be only wash. So add your washing buffer here and centrifuge again. So the washing buffer passes and then you have a clear DNA or RNA that is on those columns. And then simply you add an aqueous solution, which is mostly water, and then you can elute the DNA from these columns into a collection tube. So that saves you a lot of time, but the concept is the same. And I'm sure nowadays nobody is using like the phenol chloroform extraction to extract DNA. People are using these columns. And these protocols, from time to time, they become more and more complicated and they become more updated. So this is a paper from this year, which is showing you, in case you are isolating DNA from the cytosol, you might have a contamination from some DNA that comes from the mitochondria or some DNA from the nucleus. So this protocol is made mainly to extract DNA from cytosol, and this is in conditions where you work with viruses, where you introduce the DNA into the cytosol. I'm not going to ask you about the details, but if somebody is interested to know more, this is one of the references you can check. So they are using a different lysis buffer. It contains digitonin, which will lyse the cells very softly. And then you can, if you look at now, if you understand the details I explained for you, you can easily say, look, in this step, they are simply lysing the cells, and they are differentiating the fraction into two fractions. One contain RNA, one contain DNA, or one contain DNA and the rest of the components. Then they are adding an RNase cocktail, so they are destroying RNA. And then they are doing the extraction of the DNA by using a column. So the step I explained to you before, simply passing this solution in a column that retains only DNA, and then you wash and get it out. And then you can use it in whatever experiments. So at least when you get into those, you know those details, you are able to crack down any complex experiment, and you would understand what exactly happens. Now, I also told you that in the beginning of the lecture, I focused on extracting either the DNA or the RNA, and I don't care about the rest. But nowadays, companies are able to provide us with columns containing these cotropic salts that will retain RNA, DNA, and protein. And obviously, these are very expensive. So it depends for you. Do you really want to collect DNA, RNA, and protein from the same cells, or you just care about the RNA or the proteins? But in case you want to do this, these silica that comes in those columns contain the salts that allow DNA, RNA, and proteins to be isolated. But the way it works is that it has to have in the lysis buffer an inactivation for RNases, phosphatases, proteases, and DNases. So you want to block all the enzymes that kill RNA, kill protein phosphorylation, or kill protein itself, or kill DNA. So you want to preserve RNA, DNA, and proteins. So that's why you have a mixture of blockers for RNases, phosphatases, proteases, and DNases. That mixture will save RNA, protein, and DNA. Now, if you pass it through the column, the way it works, DNA and RNA would bind, and that's mostly because they have the guanidinium hydrochloride, while the protein will pass through the column. And now automatically you have two parts. The flow through, it contains the proteins, where the column contains DNA and RNA attached to the column, to the silica. So if you want to kill RNA, now you can simply add RNases and then elute the DNA, or the other way around. You can simply kill DNA and collect the RNA. And in this situation, you can have DNA, RNA, and proteins collected. So for more information on this, you have Takara, which is one of the biggest bio supplier for these types of kits. And there's the explanation for you with more text of what I explained. Now, in case you want to isolate only RNA, you have to consider the same thing, what we explained in the first slide for DNA, but here the steps has to be a little bit different. So in other words, you want to lyse the cells, yes, but then you have to be very careful about inhibiting RNases because RNases are so stable, you know that. And since they are so stable, they can function at different conditions. So you want to have the RNases on board from the beginning, RNase inhibitors from the beginning on board in the process. Then you want to degrade DNA and proteins, and then separate the RNA fraction. You have to make sure that you have the acidic phenol, and the same steps go for RNA from this way all the way: precipitation, washing, and resuspension. Now, the most used reagent for RNA isolation is triazole or thiazole. So there's multiple names, but they are exactly the same, which is the phenol chloroform with the guanidinium thiocyanate or hydrochloride. So whenever you work with RNA, you have to make good consideration that RNA are very prone for degradation, mainly by the RNases. So you have to protect the RNA from the first steps. Some protocols would use proteinase K, others would use the guanidinium thiocyanate. So in some protocols you find one, and others you find both. And you make sure that you are having an acidic condition so that the RNA is soluble where DNA is collected in the organic phase. And if there are any remnants of the DNA in the RNA fraction, we can add DNAs and digest that either on the column or in the solution. So if I go back a little bit onto this slide, I went a little bit quickly here. So when we have the column contains DNA and RNA, so the DNA is bound to the silica membrane, right? In case you want to do the DNA purification, you simply wash and elute the DNA. And now you go to another step, right? If you want to collect the DNA, you do the digestion of RNases here on this column, and then you collect the DNA. If you want to collect the RNA, what you have to do, you add DNAs. So you kill the DNA and then you collect RNA. And that's why I wrote this step here. In case you are isolating the RNA and you end up have some contamination of DNA, you simply use DNAs and you kill the rest of the DNA in the sample. And back to your colleague's question, if you see this is a protocol where you use the chiazole or the triazole, which is the phenol chloroform commercial name, you lyse the cells, you use this protocol which separates RNA to the aqueous solution. So in this type of protocol, you already know that it's an acidic chloroform, a phenol chloroform. You precipitate the RNA and you do a single wash with ethanol, and then you get the DNA pellet. Now, downstream experiments depends on the quality of what RNA you get here, how pure it is and how intact it is. And I will come into this in the next 2 slides. In order to optimize the extraction process, you can add those steps. So instead of doing only one time of the chiazole reagent, you can do this 2 times. And in case of washing only one time, you can wash it 3 times. And that for sure will provide you with a better quality of the RNA. And don't worry about these steps, because whenever you get a protocol in any lab, they will tell you optional steps, meaning they will say these 5 steps are mandatory. And it's written how to perform them. But they will tell you, for example, it's optional if you want to repeat this 2 times. And it's optional if you want to wash for 3 times. My experience is that most of these optional steps, if you perform them, you will have a better quality of the extracted material. Okay. could you please explain the selective elimination of DNA RNA protein you just mentioned again? Okay. So if we want to do a parallel extraction of the DNA, RNA, and proteins from the same sample. Why this is used? Because this is a powerful way if you have a little sample, say very few number of cells, and you don't want to lose the ability to check their DNA or RNA or proteins, you want to collect everything. Then you want to use these expensive columns. So the first step is to homogenize the cells with the lysis buffer that comes with the kit or which is usable for your cell type. And after that, you will take all those lysates and you will load them onto a column. This column has a property which contains these chaotropic salts, and mainly this is guanidinium thiosulfide. Because this allows the binding of the DNA and the RNA, but it doesn't allow the proteins to bind. And therefore, if you load the samples on the column and centrifuge, the proteins will pass through the column while DNA and RNA will bind. Now you have the fraction where you have the proteins. All you need is just to precipitate it. And here's how you precipitate the protein with methadone or acetonitrile. Or you have that component on the column which contains DNA and RNA. So in order to get the DNA out, you simply degrade the RNA or the other way around, and then you wash and then you elute. Okay. So after isolation of DNA or RNA, you want to check the quality and the purity of the DNA and RNA. And I'm sure any lab you go to do your projects in the future, you will see one of these nanodrop machines or the older spectrophotometers with the cuvettes where you can add the DNA and water in a cuvette and you put it in a machine that reads the diffraction of the absorption of the UV light. So the way it works, like these spectrophotometers will measure the amount of UV light that is absorbed by the nucleic acids. And it's known for us that the DNA and nucleic acid in general, like RNA too, they absorb light at the peak of 260. In other words, if you have the more the light you have at the 260 nanometer, the more RNA and DNA you have in your tube. Because if you apply UV light and it passes through the tube, there is no diffraction, then you don't have a peak, then it's mostly water. The more you have RNA and DNA, the more there a peak of light absorption at the 260 nanometers. Now, the first distinctive parameter we have to take into consideration is that proteins absorb light mostly at 280 nanometers. So if I give you two samples right now, two tubes, and I tell you which one is protein, which one is DNA, if you directly go to a spectrophotometer and you look at the peaks, the peak that comes mostly at 260 is most probably DNA, and the peak that comes at 280 is most probably protein. But this is not 100% exclusive because we will see the mixing can make a lot of changes. So to assess the quality and the purity of the DNA and know the concentration, we have to look at the ratio of optical density at 280 and at 260. While now, I don't remember how exactly this is calculated, but the absorbance of one means you have 15 micrograms per ml of DNA. That's because it's a double-stranded, whereas absorbance of one would mean you have a 40 microgram per ml of RNA. So this is how the measurement units are. The more absorbances, the more the concentration. And then if you look at the 260 to the 280 ratio, you will know the purity of your sample, how much nucleic acid compared to proteins. And obviously, when you go to those nanodrops I showed here, they have programs specialized for measuring RNA. So you have to select that you are measuring RNA as a nucleic acid or DNA. So you have to make sure that you are doing the right selection if you are isolating RNA or DNA. Now, when you perform the measurement, it will give you the values for the 260 peak and the 280 peak and the 230 peak. So in this slide, let's just look at the 260 to 280 because it will tell us the purity of the nucleic acid compared to having protein or phenol impurities. Generally speaking, this has to be more than 1.8. So 260 to 280 value, when the nanodrop gives you the values, it's going to give you the ratio right away. If the ratio is much lower than 1.8, you have to be careful because it means that if it's the 280 ratio is high, then there is a lot of impurities and therefore the value will be much lower. Now, when you do the RNA samples, considering anything below 2, I would say 1.8, then would tell you that your RNA sample contain impurities. Now, obviously, these numbers you have to memorize when you go to a nanodrop, and you have to remember that you have to look at these ratios in order to get a good DNA or RNA quality. But let's imagine we have this field here where we ran 3 different samples into a spectrophotometer. So the first thing you notice, if you ran phenol only, I give you a sample and it contains only phenol, it will give rise to a spectra that looks like the DNA. However, the peak is not 100% at the 260. It's a little bit shifted toward the 280. I would say it's around 270, somewhere like that. So in case you have a contamination of phenol that you carried out during the process of your extraction, that's how the sample will look like. You will start with somehow a high absorbance unit. So you remember the 1 absorbance unit means 50 microgram per ml for a DNA. So in this condition here, you are starting around 4. And when you measure the DNA sample, it looked like it has a peak, but it's not really at the 260 peak. It's somehow shifted to the right, toward the 280. Whenever this happens, you should directly think about having phenol that is still carried out during the process of extraction. For some reason, when you are taking the sample, when you are separating the aqueous phase from the organic phase, you carried some phenol with you. And that was the reason why you have a shift in the graph toward the 280. Now, if you repeat the steps that your colleague asked, if you go back and you repeat the steps and you purify it, then your DNA is going to look like the green one. The peak will look like 100% at the 60, and the top part of the peak is higher than the start from the y-axis. So 2 things all the time you have to look at. Where does the curve start at? If it starts very high, you have to be careful. And the other thing is the shift in that curve toward the 280 does it have more, does it have proteins or phenols, which makes the reason that the shift will take place. And this would be a typical pure DNA curve. Okay, so I wrote down here one of the examples. If the 260 to 280 is low, it indicates the presence of proteins or phenols, because the lower the ratio, the higher the 280 value, right? Because you are dividing 260 to 280. And therefore, it means that there is a contaminant that shows up there. There's one other scenario where you have to be careful. If the concentration of the DNA is very low, these three curves will be very condensed on top of each other. And therefore, you're not going to be able to differentiate if this is really a low concentration of the DNA or it is a contamination of the DNA. Question. Can you please repeat what it means when it starts high? Yes. So I'm saying every time you have a nanodrop graph or spectrophotometer that you obtain, directly look where the start of that peak compared to the top of the peak. Because if it starts way higher than the higher part of the curve here at the 260 to 80, it will directly tell you there is some sort of contamination that's taken place. In this particular example, we know that phenol starts high and it does a shift. And so in this blue one particularly, we know that there is a contaminant, but this contaminant is most probably it's proteins or phenols, because I have a shift from the 260 toward the 270 to get the highest side of the peak. But it could be something else. So in general, anytime you start with a very high curve from the y-axis there, be careful that you have some sort of contamination in your samples. I'll give you another example in the next slide, which here we are discussing the 260, 230. Now, we know that phenols come at 280, but there are many other salts that we use might come in the 230 range. So they might come in this area here. And that's why I'm saying anytime you start from a high point there, higher than the top of your curve, be careful. It would mean that you have a contamination at the 230 range. And that comes from many things. Could be carbohydrates, could be phenols, could be guanidine salt, could be glycogen that you have used throughout the protocol. So every time you have, not only look at the 260 to 80 ratio, you have to also look at the 260 to 30 ratio. Because if you ignore this, you might end up having a lot of these contaminants in your extraction, and that could lead to destroy your experiment. Now, that being said, whenever you are looking at the 260 to 230, the range should be much higher than the one of 260 to 80. It should be like 2 to 2.2. Anything lower than that, you have to be careful. And here is an example that I'm showing you in the change of the peaks. So you can directly now look at C, and you can say that this is contaminated with phenol, because two properties. One property, it starts much higher than the top of the peak, and the second one, the peak is at 270, closer to 280. So this is a typical phenol contamination. In red, you have a very nice curve where it starts lower than the top of the curve, and the peak is 100% at 260. So that is a DNA that is not contaminated. It's the A. And B in green, you have a curve that starts higher than the top of the peak. And this is typically guanidine. So as per today, with the slide information I gave you and the protocols, I'm sure any protocol that comes in your hand with the steps to extract RNA or DNA and check the quality of the DNA and RNA under the spectrophotometer, you will be able for sure to tell if the protocol is suited for RNA extraction or DNA extraction, and whether you will get a good quality RNA or good quality DNA or not. More information for you if you want to know how spectrophotometers are made. There is a link for you here. And if you want to read more about these curves and the troubleshooting, there is also a PDF for you here to read more. Now, to visualize the DNA, not under a spectrophotometer, just to check it like with the machine, you can visualize it by putting it on a gel and running. I didn't explain yet the technique, but the idea is that you would have the DNA, which is the negative charge, stained with ethidium bromide, which is a compound that intercalate the DNA. And once we hit the DNA with UV light, it will generate these bands. You can see the DNA under the UV light. Now, I will explain this in the next couple of lectures more. But the idea is, why do we need these techniques? Like the nanodrop and the spectrophotometer can give you a quality of the concentration and the purity, but the electrophoresis and running the gel, the DNA on a gel where it can be visualized, it can give you a good value of if the DNA is degraded or not. So for example, if you look at these samples 1 to 5 and 6 to 10, automatically you know that the DNA is intact here because it makes one very nice band at the same molecular weight. However, the samples 6 to 10, they are smears because the DNA has been degraded into multiple molecular weights and all of them light on the gel, and you directly know that your DNA on 6 to 10, you cannot use it. Okay, I think time's up. And I will stop here. Probably in the next lecture, we can add more. Would you mind staying back? I will be staying back. If anybody has questions, please stay, and I will stop the recording now.